Hello and welcome to the Forensic Chemistry of Hair Analysis Lab. By the end of this lab, you should be able to observe and identify different types of hair, understand the anatomy of hair, understand how hair can be used as evidence, be able to properly use a microscope, and be able to determine if a suspect's hair matches one found at a crime scene. Why is hair important when it comes to evidence? Hair has many characteristics that make it extremely valuable when other evidence has been compromised or isn't available at all. Hair is a pretty stable piece of evidence as it remains chemically and physically unchanged in conditions that would destroy or degrade other types of evidence. DNA and blood can degrade if exposed to conditions such as chemicals used to clean a crime scene. Tire tracks and footprints can disappear over time. A single piece of hair may give us information such as race, sex, and possibly the age of the source of the sample, but it is not specific evidence such as DNA or fingerprints. DNA and fingerprints are unique to every individual. So what exactly is the structure or anatomy of hair? Hair, along with fingernails, toenails, and the outer layer of skin, are composed of a protein called keratin. Hair is generally composed of three layers: the cuticle, the cortex, and the medulla. The outermost layer, the cuticle, is composed of hard scales that overlap each other and point toward the tip of the hair. The next layer, the cortex, has pigment containing granules that are responsible for hair color. The innermost layer, the medulla, is a hollow tube that runs along the hair. Basic hair types consist of human hair, where different races have different textures, and animal hair such as dog, cat, or deer hair. Basic hair types consist of human hair, where different races have different textures, and animal hair such as dog, cat, or deer hair. Hair is a general characteristic of mammals, so investigators must first determine if the hair is a human hair. Hair-forming layers can distinguish characteristics that aid the investigator in the identification of a hair type. As you can see in the picture, the cuticle, the outside portion of the hair, and the medulla, the inside portion of the hair, from a human differ greatly from that of other mammals in the picture. The cuticle forms overlapping scales that make a certain pattern depending on the species. These scales help investigators classify the hair. The medulla may or may not be present in hair. When it is present, it also aids in determining the type of hair. The medulla can be continuous and run the entire length of the hair. It can also be interrupted, which means it runs the entire length of the hair but has separations before continuing again. The medulla is sometimes not present at all. The medulla can also be fragmented, which is not pictured here but looks much the same as the interrupted pattern but has more variations in the interruptions. If hair is determined to be human, it may also exhibit certain characteristics that can aid investigators in racial determination. Asian hair is generally straight and coarse and contains larger pigment granules than Caucasian hair. The cuticle layer is thicker than both Caucasian and African. It also has the widest diameter. Caucasian hair is usually straight or wavy, ranging from blonde to brown to black, and are usually fine or medium coarseness. Caucasian hairs often either lack or contain a fragmented medulla. African hair is often curly or wavy in appearance. African hairs contain the largest pigment granules of all three types of hair. The medulla is either lacking or fragmented in African hair. The hair in the picture to the right has not been dyed. The picture on the left is a micrograph of hair that has been dyed. Can you see the difference? The hair on the right shows medulla in each hair. The hair in the middle is a picture of a blonde hair, and the medulla is more difficult to see. The cortex is the pigment portion of hair. When hair is dyed, the cortex is also dyed, and the medulla is not easy to see even under a microscope. At this time, we'll begin our laboratory experiment.